Welcome everyone to the Powerhouse Remodel, the podcast where we break down cutting edge science and debate what truly changes under our skin. I'm Adrian.
And I am Alejandro. Today we will discuss a fascinating paper published in Altan Physiology by adolescents and colleges. A project won to 20 were of supervision and network SFC, trying to alter the mitochondrial function and fusion machinery in musculoskeletal muscle.
Right, but instead of just summarizing the paper, Alejandro and I want to dive straight into a crucial argument. The main finding here is that chronic exercise promotes a pro-elongation phenotype, meaning it leads a more fused, integrated tubular network of the mitochondria, while reducing fiction of proteins like FIS1 and the mitophagy marker parking. My biggest takeaways and my argument here is that this paper changed how we look at metabolic diseases like type 2 diabetes and suggests that insulin resistance isn't about mitochondrial volume or density, but fundamentally about mitochondrial architecture.
Thanks, and sorry your point, Iron. Buen se me change 20 or the likelihood of having expectancy, to mention time where the normally center diet to circulation activity. When represented actual mitochondrial content, the child infusion protein like MFN1 and OPA1, what is artificially significant as how I was the relation infusion and mitography and happen independently of the content. So network story 1.0 allow the fusion activity in sequence or the push shopping the rising process.
That's exactly where we're looking, and other primary sources help us find answers. If we look at previous literature, for instance, a classic study by Hollis and Coley in 1984 established that endurance exercise heavily increased mitochondrial enzymes content. But if we bring more recent literature, with Gomes in 2011 in Nature Cell Biology, they found that elongated mitochondrial networks actually protect the cells against degradation during metabolic stress. This backs Alibro's conclusion by keeping a network elongated and reduce a friction FIS1, the muscle becomes vastly more efficient and discarding the glucose, which explains the 59% correlation with improvement glucose disposal rate found this week study.
I saw your point, and speaking of the primary sector, I suffer for student regulation for happen after version survey. Interestingly, a paper by the some hectares about a high inserting therapy authority and surveying function and mitochondria to create all the creation of the immunology. I will paper look as the fighting working the attention is so well or says at the negative vinegar. A worsening fry acquisitions the cause of fermenting to the clean house push long-term trying dirty and sorry residing network from resisting insulin for evolution.
Exactly. My main inquiry and lingering question about this study, though, is the sample size and population. They use 10 sedentary obsessed pre-diabetic older adults, while clinical improvements like in BMI, fasting glucose, and insulin sensitivity remarkable that we were perfectly extrapolate this exact like diamond and S1 embarking the suppression younger population or different metabolic phenotypes?
Thanks, what vital limitation. Orton associated the conversation. Actually, the local boys you were under, I will say, and noted a basically carotid control marketing different one. However, that therapy potential to consultancy, reality, I want matter exercise action no pharmacologic remodeling all the surround machinery is literal some example of the organization type of diabetes.
Well, say it turns out that fix the metabolisms, we don't just need more powerhouse, we need them to be better connected.
Beautifully point, Adrian. Thanks for today's episode. Loads of comments in your saying our mitochondrial dynamic.
Okay, thank you. See you next time. Bye bye.
Bye bye.